All right, hello everyone. My name is James Newberger, and today I'll be talking about the targeted inhibition of the WRN helicase, replication stress, and cancer. This topic is keeping in line with our theme for the year of papers about cancer, and we're going to be learning a little bit about what the WRN helicase has to do with cancer. All right, we're going to start with a little bit of an introduction. So WRN helicase, that is absolutely the star of this paper. And for good reason. There are several roles of WRN helicase, such as replication, base excision repair, recombination, DNA damage response, transcription, and it's an early enhancer of the RNA polymerase 3. As you can see, there's a little picture of the WRN helicase. It may not look too complicated, but it is very important to cells. And it's especially important in cancerous cells because in those cells, it prevents the mitotic catastrophe, prevents replication stress, keeps up cell damage repair, and assists in the fork progression for proliferation. Now the highs and the lows. So there are different levels of WRN helicase. As with anything in the body, it can be raised and lowered. And it has been found that high levels of the WRN enhanced cancer cell cycles, increased the cancerous cell proliferation, and induced cell repair overriding. So what that means is in these cancer cells, it was both enhancing the cell cycle to proliferate faster, and it was overriding the repair mechanisms built into the cell to stop these cells from growing, and overriding that by making sure that it kept growing and stopping the cell damage stress. It has also been found that in low levels of the WRN helicase, there are problems with translesions, stalled DNA forks, genome fragility, and cell death. So if a person in general has low levels of the WRN helicase, it is really bad for them. If that's like spread throughout their body, it can be caused by Werner syndrome, which is something we talked about in class. And some results of having this would be neoplasia, malignant melanoma, and soft tissue sarcoma. So it's bad to have high levels of the WRN helicase, but it's also bad to have low levels. So with anything, it's good to be in equilibrium with the WRN helicase. Next, we're going to be looking a little bit at the contents of this paper. So relating to what we know. So this is stuff that we already kind of talked about in relation to helicases in general. So the functions of the WRN helicase are DNA strand displacement, proofreading and base excision repair via the DNA helicase, and then 3' to 5' DNA exonucleotic activities. So looking at this little graph over here, you can see that the WRN nuclease, which it looks like a little bit of a kidney bean, can be found in the base excision repair step and the proofreading step in the replication fork. So we already know that the helicase is important for helping to unwind, helping to unzip, but it does a little bit more than that as well. Then, adding to our knowledge a little bit, the WRN helicase operates in DNA damage response in the absence of operative Fanconi anemia pathway. So what this is saying is when there's the absence of damage response in the cell, when the cell has been programmed not to repair itself because it knows that it's a bad cell, like a cancerous cell, the WRN helicase will then step up and respond to the damage itself. It has been found that low levels of the helicase nuclease, due to targeted inhibition, lead to replication stress owing to translesions, stalled DNA forks, genome fragility, and all of those trigger cell death. So what that is saying is that when there are low levels of the WRN helicase within the nucleus, within the cell of the cancer cells, then the cell could be destroyed It could deal, or it couldn't deal with the replication stress, and the DNA fork would stall. So what that means for us is targeting those WRN helicase DNA segments within the cells that have cancer would be a good way to stall those replication forks and cause more replication stress. So now resisting the WRN. So looking at the paper, they found that two small molecule inhibitors, NSC 1963-0 and then NS 617145, were available against the human WRN helicase. They used these two inhibitors for specific targeting of the WRN within the cancerous cells, and they found that the NSC19, I'm going to call it that just for short, it impaired the response of the treated cells to DNA damage and replication stress. So the NSC19 focused specifically on stopping the WRN helicase from responding to that DNA damage and the replication stress, causing those cells to die. They also found that the NSC19 caused acute cytotoxicity in the normal cells, and then in the primary mesenchymal cells. So they found that the cytotoxicity caused the cells that they were targeting to die, obviously, which was a good thing. They wanted those cells to die. So that was a good find that they had. And then in the NSC61, they found that it cooperated with low doses of mitomycin C in restricting the proliferation of the cells that they were looking at. So they found that in addition to killing off the cells, even if that didn't specifically work, the NSC61 cooperated with the mitomycin in stopping the cells from expanding out further and proliferating. So these two in conjunction both killed the cells that they were targeting and stopped those cells from continuing to grow and proliferate, which is huge. So this is a little bit of a look at what the targeting of the WRN helicase does. These are both figures that they had within the paper. So they found that a knockdown of the WRN by the two methods that they looked at, both using the NSC19 and the NSC61, led to a marked decrease in cell viability and mitotic catastrophe. So the figure on the right shows the three methods that they had looking at attacking the DNA within the nucleus of the cells that they were looking at killing and stopping the proliferation of. The figure on the left, it shows in the glowing dyed sections. So A, C, and E are pictures that they took, and then B, D, and F are zoomed in sections of that. And it's the same for the other side with the blue. They saw that the ones that started glowing, the luminescent ones, were the ones that had the knockdown of the WRN. And they found that it was very, very effective in the cells that they were working with, as you can see in the pictures, because all those cells are glowing, and that's huge because the WRN levels were lowered, which again leads to the decrease in cell viability and the mitotic catastrophe, which is great again, because that means that the cells that they were targeting specifically were incapable of growing and proliferating, and those cells were the cancerous ones. So in conclusion here, we can inhibit cancer proliferation and growth. While these may not be extremely usable methods yet, because they're in the early stages of testing, in the future, using the EGS, RNase-P, and the RNA interference targeting systems could be further advanced for fighting cancer by exploiting that capability of tumor cells to overcome cell cycle checkpoints under conditions of deficient DNA repair and fork progression. So in targeting that WRN helicase, in stopping that repair, stopping that proliferation, they were able to kill off the cancer cells. And then these targeting systems utilize degradable RNAs that are not supposed to apply to a long-term selective pressure on cancer cells and can direct these cells to cease synthesis of DNA and proliferation. So by using those degradable RNAs, they could apply, like it says, long-term selective pressure on just those cells. So by focusing it on the cancer cells, it would degrade before it got beyond them. And that meant that they could use multiple rounds of this treatment on the cancer cells without harming the surrounding environment. Which, as I keep saying, that's huge, because that both means that the cancer cells can be targeted themselves without the risk of harming the cells outside of that enclosed system, which is amazing. And that is it. I hope you enjoyed listening to me talk about what happened within this paper, and I hope you have a great summer. Thank you.